Okay. Anybody got any questions for me? Ferrets. Yeah. I had a ferret. Well, I didn't have it. A girlfriend of mine had a ferret one time. I wasn't real crazy about it. And they're okay. They're neat. But all I remember was it got into a lot of stuff. It would climb underneath my kitchen counters at night because the doors didn't close very well. I would bat at it till it could open them, and then would run around through the pots and pans at 3 o'clock in the morning. I don't exactly know, Emily. Yeah, I know the one you're talking about, the phosphate that gets attached. Yeah. All of those phosphates on ATP are called inorganic phosphates. The little I indicates that. But honestly, chemically, inorganic implies to me that it's a phosphate without carbons. And if you look at that phosphate, those phosphate groups, am I near that slide? I am, okay. Yeah, here we go. They don't have carbon, but they don't have hydrogen either. So, again, I'm not really sure exactly what that means. I'm sorry. I mean, I know what organic and inorganic means. I'm just not sure what it means in reference to the phosphate there. Any of you guys had chemistry lately and knows what it means? No, any idea what that means? All right, remember you have your exam next week, and that's going to include chapters 4, 5, and 6. We'll finish chapter 6 today. I'm not going to go into chapter 7, only because it's a relatively long. It's a relatively long chapter, and you got enough to think about with your exam coming up. This is a good chance to really do well on the exam. There's not a lot of information on it. Well, there's not a lot of stuff that we've gone over. There's a lot of information on the exam, don't get me wrong. But, you know, you should have a chance perhaps to pull up your grade, especially if you didn't do as well on the first exam as you like. And we were talking before class about possible ideas for essay questions. And I said that I had actually kind of said something about that in class the other day when I mentioned compare and contrast questions. And there's actually a couple of spots in here where I could ask some neat compare and contrast questions. The way things move across the membrane, and we spent a lot of time talking about that, active and passive, carrier proteins. Like if you think about a compare and contrast question, what I'm looking for again is how are they the same? How are they alike? All right, like for example, if we were to say what's one of the similarities between active transport and passive transport? Carrier proteins, right? They both have carrier proteins. In active, it's a pump and pumps things uphill, but in passive, we had facilitated and protein channels, and those are both carrier proteins. They just happen to move things downhill. So, you know, and we could also do that with some of the stuff we're going to talk about today, which, by the way, you guys are going to have to help me remember. I think that's where we finished, right? In my notes, I have that, but I can't remember if we went beyond this slide. Yep. Okay, good. That's what I thought. Thank you. I have two sections running at the same time. You guys are just a little bit off of each other, so I don't want to make sure I just don't jump up and everybody's like, wait, we haven't talked about that stuff yet. All right, so again, this whole chapter in chapter 6 is about metabolism. It's about energy. It's about how living systems bring in a source of energy, and we have to bring it in. We can't make it, remember. We can't. Even the plants don't make energy. They photosynthesize, but they're not making energy. They're obeying the laws of thermodynamics, that first law, which says energy has to be converted. So again, the amount of energy in the universe, whatever that quantity is, and my guess is physicists have come up with something like that. Whatever that X amount is the amount it's always been, the amount it always will be. It's just spread out. Okay, it's just spread out differently. So if you need energy, and living things, we need energy because we don't want, because the second law of thermodynamics says things fall apart, right? Things fall apart. They do. Think about some old piece of farm machinery that might be sitting out on some property that nobody uses anymore. Throughout the ages, the paint comes off and then it rusts, and then it structurally begins to fall apart. And maybe in 200 years, maybe in 100 years, depending on where you are, the only evidence might be someone who could dig up the dirt and realize there's a lot of iron in there. Yep, 4. That's it. 4. 47 is what I should say, which was, what, isn't that the answer to Hitchhiker's Guide to the Galaxy? The answer to life is 47, or was it 42? I can't remember. All right, but so everything falling apart. So the second law says everything falls apart, can't do anything about that, but you can kind of help. Yeah, I think it was 42. I always remember 7 times 6. You can help not falling apart quickly by using energy, because energy allows you to maintain order. Energy allows you to maintain order, allows you to maintain homeostasis. That's all the same thing, folks. Staying in homeostasis, right? Staying in balance. And remember the way our bodies work, it's going to be kind of the way you drive. You're not exactly at 47.5 miles per hour. You might be 45, 49. So you have this average balance. So your body's doing the seesaw thing back and forth. You don't want to get too wild, of course. But that's balance. That's order. In order to do that, you got to have energy. What enzymes do, which is what we're going to get into now, is enzymes allow living organisms, remember, to get a reaction, a chemical reaction, a necessary metabolic chemical reaction, but cheaper. That's exactly what they do. Let me go back one more. There we go. So when we looked at this chart the other day, if you go from what we start with, the reactants, in enzyme terminology, the reactants would be the substrate. Okay, reactants is more general term. And then those reactants are being turned into products, and you can see what has happened in this particular case is energy has been transferred, right? The progress of the reaction you can see on the y-axis, and there's energy. What the little hills are, remember, are energy of activation. It's like priming the pump. It's like I said, taking a gallon of, well, even in the internal combustion engine of your car, in the piston chamber or in the cylinder chamber, when the gas is put into the chamber, it still needs the spark plug to set it off, doesn't it? Gas will burn, yes, of course it will, but the spark. So think about the energy of activation maybe is kind of being the spark, the little bit of energy that's necessary to get it to go. What enzymes are going to do are they're going to allow you to get the same reaction, less spark, less energy out. So it's like investing. You got to invest a little to make a little. Well, if you were to invest a dollar and get back $1,000, that would be nice. But if you could invest $1 and more efficiently make $10,000, well, that would be a benefit, wouldn't it? So that's what enzymes do. Enzymes give us the red curve. They allow us the reactions that we need to stay alive to occur at a cheaper rate, so we can conserve energy. And we can allow reactions to occur that wouldn't normally be able to happen in living organisms because of the need for typically some temperature extreme that we wouldn't be able to deal with. You can tear starch apart by boiling it, for example, but boiling is not really an option for us if we want to separate starch molecules into little glucose, so we can use that. All right, now as we said in class the other day, the thing about an enzyme, it works like a lock and key, right? My key won't open up the door to your car, and we've talked about this a million times. And we've also talked about the fact that enzymes, which are proteins, and proteins have shape. And in that enzyme, a particular area of that enzyme has a particular shape, that area called the active site. And the reason why that shape is so particular is because an enzyme is specific for its substrate. So enzyme A will only work on substrate A. All right, now I want to make sure that you understand this terminology before it gets confusing, because I see it on the exam sometimes. By definition, an enzyme has a substrate. If you're a substrate, it's because you bind to an enzyme. There are lots of other molecules out there in the cellular environment. Lots of them. Some of them are substrates for other enzymes. But you can't have a substrate that is specific for one enzyme. It won't bind to you. All right? So in other words, what I'm saying here is when we use the word substrate, we're talking about something that binds to a particular enzyme. Because here in a few minutes, I'm going to talk about other molecules that can get into the active site. You see, there are other molecules that can bind to the active site, and they do certain things that are important, but they wouldn't be called substrates. Because by definition, a substrate is going to be turned into something. It's going to be turned into a product, okay? So more on that later on. Now, organic catalysts. Chemical or enzymes are big, huge organic molecules, and they're catalysts. What a catalyst does, we define as, a catalyst spurs a reaction, so like you would do a horse, spurring a horse. So it spurs a reaction, or it It edges a reaction or gets a reaction to go, but it itself, whatever is the catalyst, does not get changed. Think about that for a second before we go on. It does not get changed. All right, so the enzyme takes substrate and turns it into a product, but the enzyme itself isn't changed. Nothing happens to it. Why do you think it's important, or why do you think that was done among living organisms? Why are enzymes reusable? Why would you think that? I mean, yes, they have an important function. I understand that. But I'm talking about you could accomplish that important function with an enzyme. You build it once, it turns the substrate into a product, and then the enzyme falls apart. Why does that not happen with enzymes? Why do they keep going? Why can an enzyme be reused over and over again? Does anybody have? And again, don't make this more difficult than it really is. Yeah, of course it is, right? It's more efficient. Think about, I told you before, enzymes are expensive. And what I mean by that, of course, is there's a lot of molecules, a lot of atoms, a lot of bonds that require energy. So enzymes are expensive to build. I could use a really good example. Imagine if when we went and bought cars, right, the money that you spent, they only lasted once. I go buy a car, I drive home, whoop, that's it. No more car. That's a heck of a lot of money to pay for something that only lasts once. So an enzyme's kind of like that. It's kind of like a car, right? It's going to want to be used over and over. Now, before we go on, will an enzyme last forever? Will it always be able to do its job? Why? Yep, it's going to fall apart. You got it. Just like your car is going to fall apart. That enzyme, through successive uses, the protein, the shape, it's going to shift a little bit. It's going to get a little dinged up. Whatever you want to say, it's going to come to a point where that enzyme no longer can do its function, and then the cell's going to tear it down, right? And use all those little amino acids that are in there, right? Make another protein. Isn't that great? It's like Legos. We talked about the other day. Dorian said she liked Legos. I did too. You can build anything with Legos. You tear down those little blocks and you can build something else. It's the same thing. It doesn't mean that the enzyme, you've wasted all of this this material. You got a lot of use out of that enzyme, right? And then when it's done, you can recycle it. I didn't build anything creative. I just built houses, but I thought that was kind of cool. Now, on the right-hand side, the induced fit model always reminds me that Mother Nature is not perfect. And what that means, and this probably... yeah, that's why I don't like the ones that are already have the Star Wars ones. I like the ones that you do your own thing. So as this enzyme is doing its job, maybe it was made just a little bit different, so the active site is just a little off. Or, like we said earlier, maybe it's getting old and it's wearing down. When your tires have wear on them, you don't got to replace them until a particular point. Still got wear. So maybe that enzyme is getting worn down through time. So this induced fit model tells us that enzymes will accommodate, or enzymes and substrates binding, will accommodate little minor changes that might occur in the active site. There is a limit, of course. You eventually do have to change your tires. But the induced fit model is kind of Mother Nature's way with enzymes of saying close enough. And if you think about that, that goes back to the efficiency thing again. You wouldn't get rid of your car or your tires, let's say, if they were only half worn down, would you? Right? You would try to get as much life out of them as you possibly can. So as that enzyme begins to wear down and suffer entropy and minor little shape changes, as long as they can still induced fit, as long as it can get close, it's still going to work. I love that about Mother Nature because we see it later on in genetics, actually, where Mother Nature always kind of builds in, living systems have always built in a little bit of a fudge factor, just to say, well, we know things aren't always perfect, so let's not waste this entire enzyme because the tires are only half bald. Does that make sense? Think about it, enzymes are really cool. This is a neat enzyme. Any questions on what I was just saying? Where's my pencil? No, everybody good? This is an enzyme. This is, and as you can see, it's pretty complex. Enzymes are fairly complex, but what I really look at is the name. And I don't really need you to know the name for any reason, but there's a couple of little things in the name of an enzyme very often, not in every enzyme, some have pretty funky names, but many times it can tell you that, number one, it is an enzyme that's being talked about, and number two, even what it does or what its substrate is. See, enzymes are specific for their substrate. So, for example, sucrose, right? Table sugar, the name of its enzyme, sucrase. See, it's the ending. Remember, OSE ending usually meant what, carbs, right? Fructose, glucose, galactose, right? ASE oftentimes refers to an enzyme, like this one down here. See the ASE at the end? ASE. And to kind of stick with this idea of specificity, very often the name of the enzyme is the name of the substrate with the ASE ending put at the end of it. Not always, but very often. So now when you're looking through your book and you come across words like this one right here, pyruvate, and I don't really care if you know what pyruvate is. It's a molecule. You will know in the next few chapters because I'm going to introduce you to it. But look at the name of this enzyme. For one thing, again, it ends in ASE. This enzyme tells you what the substrate is, pyruvate, okay? And it tells you what it does to it. What does pyruvate dehydrogenase do to pyruvate? This is kind of the same thing when I said, how do we make a deoxyribose sugar? Well, maybe not dehydrates, but dehydrogens. Takes the hydrogens off of it. Okay? Takes the hydrogens off of it. Okay? Makes sense? Isn't that cool? You'll see another one, and again, we'll do the same little trick with the name. It's kind of neat. And again, I'm not concerned with you telling me, yes, you did. Pyruvate is what begins the Krebs cycle. I'm not concerned with you knowing all about pyruvate dehydrogenase, but I do want you to understand the endings and how that often indicates an enzyme, so that it helps you when you're reading some of these words in your book. And again, if you take time to not just bleep over these words, go back and say, oh, I see, and I can even figure out what it does. And you're exactly right, Tulsi. That is very important in the Krebs cycle. Now, I was talking earlier about enzymes being recycled, right? So let's take one more look at this idea, because I want to make sure that you understand that enzymes can do multiple of things. It's not necessarily one enzyme, or excuse me, one substrate turned into one product. They can do things like put substrates together. They can take substrates and split them apart. They can take substrates, put them together, and change them, like you see in the picture here. What do we got? We got a green and a red substrate up there in the upper part. It's binding to the active site. Notice that it fits in. You need both of them to fit in. So sometimes it's two puzzle pieces, and then boom, boom, in the next step, the substrate and the enzyme are doing its thing, and we're converting the green and the red into a yellow and blue product, whatever that might be, and then that's expelled, right? And the enzyme goes around and picks up another set of substrates. So down below is a nice, really easy way to keep in mind what I said about enzymes. Any enzymatic reaction, sucrose, sucrase, this one, pyruvate, dehydrogenase, always works the same way. It's an enzyme plus a substrate, okay? And that makes the enzyme substrate complex that you see. And what's released, and this is the important thing, what I mean about enzyme, what's released is the catalytic enzyme. Oh, now my pen stops. The E is released, so E goes in, E comes out. And what is changed is not the enzyme, but the substrate turned into product. Okay? So right there in that little simple sort of equation there highlights the whole idea behind the fact that the enzymes move through this cycle, continually turning substrate into product, substrate into product, substrate into product, without getting changed themselves until they fall apart, right? Until entropy reaches. How are we on that? That cool? Good place to end at the of that topic, because now I want to talk about something that we do with enzymes. It actually goes back to the first beginning of the semester. No questions? Good deal. Living organisms have to stay in balance, right? What's that word? What's that word we use to refer to living organisms being in balance, or the idea of maintaining that balance? There you go. Homeostasis. That's right. And homeostasis involves a lot of things. It involves stop, controlling, it involves go, and involves everything in between. Right? That's what I'm saying, like that balance of homeostasis in terms of chemical reactions, enzymes doing their chemical reactions. There's going to be times when you don't want those chemical reactions to occur, right? If you got enough of this product or whatever the case may be, you don't need to waste making any more. There's going to be times when you're going to want those chemical reactions to go at a really fast speed. And then, of course, everything in between is going to be necessary, because maintaining balance is not about on and off. It's about how far on. It's kind of like the volume knob on your radio. Or more, as I said yesterday, it reminds me of teaching my daughter how to drive. My daughter, who's a relatively impatient person, only understood that the accelerator was the car was not moving or going full speed. Now, I make a little bit of a joke, but I used to always tell her, Stephanie, that pedal is a variable speed device. Let off on it a little bit, you don't have to go full speed. She didn't really learn that till after a few traffic tickets, but nonetheless. So it's the same thing with enzymes, right? They can go really fast, they go really slow, and they go really someplace in between. So what we're going to talk about now is how that's regulated. How do we adjust the speed of enzymatic reactions? Okay, very simple, very simple. Let me show you how it works. Look at the far left-hand side of this picture. This is a normal enzyme and a substrate. This is what we've been talking about so far. Notice that the substrate and the active site shapes are nice. They're going to bind. Substrate's going to be turned into product. Enzyme goes about its way, right? Now in the middle, what if we had a molecule? Now I'm not going to call it the substrate. The thing in the blue, I'm not going to call it the substrate, because by definition, substrates get turned into product. So this competitive inhibitor can bind to the active site, at least enough, right? But it's not going to be turned into product. It's going to do something, though, as you see in the little red arrow. It's going to stop the substrate from binding to the active site. All right, now you put that together for me. If the substrate can't bind to the active site because the competitive inhibitor is in the way, are we going to get any product? Penny, are we going to get any product? If that substrate can't bind to the active site, are we going to get any product? Does that competitive inhibitor get changed into anything? Does it get changed into a product?
It's unusual shape.
It's not. No, it's not the substrate. That competitive inhibitor is not the substrate. Remember, by definition, substrates become products. The green, light green thing, that's the substrate. If it could get to the active site, it would become product, but now it can't. The competitive inhibitor is not going to be changed into anything. It's not binding to the enzyme to be modified. It's binding to the enzyme to inhibit the enzyme from doing its job. And it does that very simply. It gets in the way. Right? See that competitive inhibitor? It's in the way. Some competitive inhibitors fit the entire active site, but you don't really have to. Just enough to block the normal substrate, you're going to inhibit the reaction. Now, you might be thinking, gosh, you just spent how long telling me how expensive enzymes are, and now you say we bind a competitive inhibitor and the substrate can't bind. We just took the enzyme out of the loop. Seems kind of pointless, doesn't it? Things don't stay bonded forever, folks. Enzymes and substrate only make an enzyme-substrate complex for milliseconds, maybe, and then the products are released. It works the same with inhibitors, too, by the way. When a competitive inhibitor binds to the active site, it's not there forever. It stays bonded for a while, and then it disconnects. And then when that active site is open, that's why it's called competitive, the game is on. Competitive inhibition has its name from the fact that it's either the inhibitor or the substrate that get to the active site first. If the substrate gets there, product. If the inhibitor gets there, bumpkus. We don't get product. That's a stop and go part. How do we control the speed on that now? It's a numbers game. Let's see if my pen is going to be nice to me. Let's imagine an environment with some enzymes in it. Make some real simple numbers. Let's say we got 100 enzymes. Okay, and in that same environment, we have 100 substrates. So one substrate for every enzyme. Okay, this environment could be the cell. Well, it would be the cell. So in this cell with this enzyme and the substrate, there's an equal number. So you can imagine substrates binding to enzymes. Now remember something I talked about I don't know, a few weeks ago about the randomness of all this, right? Substrates and enzymes get together because they fit, but they get close enough to fit because they randomly bump into one another. Competitive inhibitors are the same thing. So when randomness occurs, right, one of the ways you can regulate that is through numbers. So back to the 100 enzymes and 100 substrates. What if we wanted to slow this reaction down, slow it down, or even bring it to a point where it's almost stopped? What we could do in the environment of the cell is the cell could create, okay, or it could bring in from the outside some competitive inhibitors, things that would bind. And let's say the cell says, I really want to slow this down, so I'm going to import, maybe through endocytosis, 100,000 competitive inhibitors. So that's what we got in the environment. 100 enzymes, 100 substrates, 100,000 competitive inhibitor molecules, all floating around, all randomly competing for the active site, all dependent on, or what happens dependent on who gets there first. Now, if you were a betting person, and I understand that a substrate can bind, but if you were a betting person, not my phone, would you bet that you would get inhibition, or would you bet that you would get substrate turned into product? What is more likely to happen with that kind of an environment? Tulsi?
If it's
Think about it again, Penny, look at it. In an environment in which there's 100 enzymes for 100 substrates, which one become product, but you've thrown in 100,000, right? 1,000 times more competitive inhibitor. If it's 1,000 times more competitive inhibitor, if you're playing probability, what do you think is going to bind to the active site? Do you think it's going to be one of those 100 substrates? Could be, yes, could be. Or do you think it's more likely it's going to be 100,000, one of those 100,000 competitive inhibitors? Is it competitive against? No, the competitive inhibition is the inhibition part. You don't get product from that. What I'm talking about here is how we regulate that. Does anybody understand what I mean by the numbers there? Do you see how looking at that the chances are is you're going to get an inhibition? Because that competitive inhibitor binds, right, the blue thing, for a while, then it unbinds, and then the race is on again, but you got 100,000 competitive inhibitors and 100 substrates. To give you a visual, imagine you reached into a bucket and there were 1,000,000 marbles, 1,000,000 green marbles, all the same color, all the same size, and I said, I'll give you $1,000 if you can pull out a red marble. There are 2 red marbles in the bucket, 1,000,000 green ones, 2 red ones. I'll bet you $1,000. You're not going to bet $1,000 you can't pull a red one out. Would you take that bet? Gosh, I'm hoping some of you would. That'd be an easy $1,000, wouldn't it? You got 1,000,000 green. You're going to close your eyes, reach in, grab a marble randomly. You're going to hope that it's one of those 2 red. It's like playing the lotto, folks. Now, if I wanted to speed this reaction up, what the cell would do is remove the competitive inhibitors, maybe pump them out of the cell, or maybe send lysosomes to get rid of them, right? Remove those. So decrease their numbers, let's say down to 5, just to be silly. Now, what do you think is going to happen? If you're going to bet now, do you think you're going to get substrate to product, or are you going to get inhibition?
Inhibition.
You get product or inhibition. Product or no product. If you're in an environment like this, chances are. Yeah, chances are, right? Why did you say that, Jordan? Why do you say the chances are that you're probably going to get product? How come? Yeah, it all fits in there, right? Yeah, thank you, Emily. There you go, same idea. There's more substrate out there. All right, so as you monkey with those numbers, and you can do it along all 3 levels. You can add more enzymes, more substrate, less substrate, more. You see, all of those are those little knobs on your stereo that you probably don't know what they do. Well, maybe you don't have that anymore, but years ago we used to have stereos, all these little knobs, and a buddy of mine used to say that half the time the only thing they affect is if you can hear like a dog. which I can't. But yeah, you got all these little switches you can go back and forth, all these little things you can turn. And that's what I mean about homeostasis not being rigid. Okay, he does. Does it still work? And not being rigid, it's always kind of a moving. You're always kind of, homeostasis is an average. We don't want to get like this, but, throughout our day, we're a little hungry. We're full. We're a little thirsty. You know what I mean? We're a little warm. We talked about this already. Does everybody understand the importance of regulating enzyme activity? You got it. Of regulating enzyme activity through numbers. You understand the number thing I just went through? Does that make sense to everybody? Does anybody need me to clarify that? Because the last one I'm going to talk, or not the last one, the second one I'm going to talk about on the end is going to involve basically the same concept. So I want to make sure that we have that down straight. Everybody's cool with that? Let me erase my script. Yeah, bigger numbers usually what's going to happen. You got it. Thank you, Emily. All right, now in the last one. As far as the numbers and the way it's regulated, same way, right? It's all going to be about the numbers. But in this time, we don't have a competitive inhibitor. We have what is it, orange? Yeah. We have an orange thing called a non-competitive inhibitor. And what the non-competitive inhibitor does, the name, it does not compete for the active site. See, it doesn't bind to the active site at all. But you see that site that we haven't been talking about? Oh, come on. There we go. Right over here. That's another binding site. The name for that binding site is called the allosteric site. It's called the allosteric site, or it's called the non-competitive inhibitor binding site. It's also described sometimes as the inhibitor binds the enzyme at a place away from the active site. So that's a little, you might see it referenced and you go, what inhibitors? That's a non-competitive inhibitor. It's binding to this other site, this allosteric site. Now, if you're wondering, well, how does this stop the reaction? Look at the active site in that picture. Is that active site the same shape as it was, say, in picture A? Or has it changed? Yeah, it's not the same, right? See this little thumb area right here? It's sticking out, isn't it? We changed the shape of the active site. Now, go back for just a second. You can change the shape of proteins. We know that. If we change the shape of the active site, will the substrate bind? Outside of the induced fit part. If we change the shape of the active site enough, will the substrate bind? Hi, Tulsi. Good to see you. Will it? Will it bind? Look at the shapes there. It's all about shape. Probably not. Yeah, probably not. No, it won't. Right? It won't bind. So if you can't get binding there, right, you're going to get no product. And of course, the same thing. If you have a lot of non-competitive inhibitors, there's a chance you won't get product. If you have fewer of them, just like we talked about before. Now, what I want to explain to you, though, is how do we get back the other way? Well, the non-competitive inhibitor doesn't stay bonded forever, just like the competitive one, falls off, and when it does, the active site goes back to the correct position. It's called non-competitive, but it actually is kind of competitive because you can never get both substrate and non-competitive inhibitor binding at the same time. In other words, the other side of this story, if the substrate was bonded into the active site, if you had the right shape, like what would happen in part A, the allosteric site, this one here, that changes shape so that inhibitor can't bind. So it is kind of competitive. Whoever gets to their, yeah, it is. Oh, Emily, that's real. Yeah. There is a type of breeding behavior in fish called sneaking. And it's kind of like what you're saying. Males who are less dominant among groups of fish, who if they try to mate with the females will get beat up by the bigger fish, hang out on the edges during breeding time, and they whip in real quick and try to breed with somebody. It's called sneaking. It came to mind when you said that. But yeah, that's kind of what they're doing. So if the substrate binds, the non-competitive inhibitor can't. If the non-competitive inhibitor binds first, I should have said first with the substrate, then the substrate cannot. Does that make sense? It's not hard, is it, right? Makes sense. This is kind of the same thing. Competitive inhibition, anyways, like the little plastic things you put in the outlets. Your kid didn't stick in a paper clip. right? It doesn't do anything, doesn't boil your water, it doesn't dry your hair, what you stuck in there, but it prevents something else, your kid, the substrate, sticking his finger in there. I never thought that was a good idea as a kid. I always kind of inherently figured that was not a good thing to do. All right, I want to talk about one more type of inhibition, but it's not different than the two we've been talking about. It's in the process that's a little bit different. So let me first start with this. As you can probably imagine, metabolic reactions can be very complex, and many of them are complex. So you can probably imagine that it's more than just an enzyme and a substrate product. We're done. It's like a conveyor system, like an assembly line system. So in this particular case, we're making, I don't know, this end product, this purple six-sided thing, whatever that might be. And we're starting at the top with that gold-colored sphere. And it takes a variety of enzymes to do this. So we get an initial substrate, an intermediate substrate, which is, of course, the substrate, or which is, of course, the product from enzyme 1. And then from enzyme 2, it turns into intermediate substrate B, which is, of course, the product from enzyme 2. But it's like not a final end product. Only when we've made it through those 4 enzymes do we get a final end product, right? It makes sense if you think about what we just kind of know about metabolic reactions. You know, they're pretty complex, and some molecules, you know, like in machine shops, if you've ever been in a machine shop, when they shape metal machinery or anything like that, you know, they do it in stages and steps. They whittle it down, they get it to the right shape, and all that sort of stuff. So the same thing is happening here. I'm not really saying anything that's really too outlandish, right? But there is a chance for inhibition in here. And this is probably, to me, one of the neatest things about biology and physiology. And if you're going on into A&P, you'll learn all about these things called feedback loops, positive and negative. So that's what I'm referring to. The final end product down there at the bottom. The key to inhibiting this set of reactions is using the final end product as the inhibitor. Well, that seems kind of silly. Why would I go through all 4 steps just to make an inhibitor? Well, you didn't make an inhibitor. You made something the cell needed. But you probably made more than the cell needed. All right, do some numbers again. Let's imagine we need 100 of these. The cell needs 100 of these for some reaction. Starts up the chain, starts up the chain, starts up the chain. Bah, bah, bah. We start turning A to B, B to C. Bah, 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 quick, and might get 10, 20, 30, 50. All right, 100. Cell says, okay, I got 100. That's all I need. Well, you see, when we reach 100 here, there's still some intermediates there. And you can't tell an enzyme to stop. It doesn't have a brain. So it's going to do what it does. It's going to bind to something that sticks to it. It's going to turn it into product. So it's what we call chemical inertia. Y'all know what inertia is, right? Give me an example of inertia. You guys ever experienced inertia? I always thought that was a cool word. I'm going to name my kid that. If I was in class, what I would do is I'd walk across the class. Okay. I would walk across the class with a chair, a rolling chair, pushing and pushing and pushing. Then I would stop and let go of the chair. What would the chair do? I'm walking, I let go of the chair. What would the chair do? I'm pushing and I'm pushing and I'm pushing. I'd stop. I let go of the chair. Yeah. No. Eventually. Not right away, right? I'm walking with the chair. I stop. I let go of the chair. Chair's on wheels. Chair does what? No, it keeps moving for a bit. Yes, it eventually stops. Yes, it eventually stops. That's inertia, right? That's inertia. You know, that's that force that you feel. Okay, so there's a chemical inertia, right? So when you get to 100 and the cell says, oh, thank you, you did 100 end products, thank you. There's these other ones that are still in the pipeline. Boom, 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 they get cranked out. So I'm getting ready to talk to you about inhibition, but specifically what I want you to focus on is not the end product itself. It's going to go off and do whatever the cell needs those 100 for, but it's the excess of that end product. It's the ones that get made beyond the 100. It's the A that gets made into B, and the B that gets made into C, and the C that gets turned into the end product beyond 100. Let's say 20. I don't know, just for a number, simple number. Maybe we end up making 120. Now the cell uses the 100 and does something with it. The 20 it doesn't need, so it sits around as excess. Now sometimes that happens and that excess just falls apart and the cell says, all right, chemical inertia, nothing I can do about it. But there is a way to regulate with that excess too. What if we could take the excess end product, not the 20 in this case, and what if we could use that as an inhibitor? And what if we were really smart and we said we're going to inhibit not the second or the third or even the fourth enzyme? Let's start up at the very beginning. If we have an assembly line of cars and we want to make 1,000 cars, where's the best place to tell the people to stop making any more cars in the assembly line? The beginning. Because if they don't do the start in the beginning, enzyme 2 or the next person in line can't do theirs. So you're going to go back this. Oh, almost grabbed the wrong pen. You're going to use this excess end product as an inhibitor, and you're going to inhibit enzyme number 1. Very often, the way you're going to inhibit enzyme number 1 is the way Emily mentioned, the sneaky way. You're going to do it through allosteric. Let me show you an example, okay? All right, up at the top, we've got something called threonine. Threonine is an amino acid, and we want to turn threonine into isoleucine, another amino acid. You'll learn, especially if you do go on an A&P, that some amino acids you can't assimilate. In other words, you eat them, but they cannot be taken up by your body. They go in and they go back out. But you still need them. What your body does to get around that is it converts one into the other. So in this particular case, in this set of reactions, we're converting one amino acid, threonine. Do we need threonine? Well, yeah, but we have enough of that. We're converting that into isoleucine. Now, that's a complex step. It takes, 5 enzymes, as you see there. Before we go into the actual inhibition part, look at enzyme number 1's name. First of all, convince yourself that, never having seen that word before, and I'm thinking you probably haven't, that you know that it's an enzyme. What's the cue that it's an enzyme? Other than the fact that it says enzyme number 1. There you go. And any idea what threonine deaminase might be doing? In other words, what is that enzyme going to do to that substrate threonine? So I said not all enzymes have their names like this, but sometimes you look at that and go, oh. Any ideas? Well, the threonine is the substrate there. Think about that as the noun, and think about deaminase as a verb, if you will. So what is the enzyme going to do to threonine? Again, this is very much similar to when I went, how do you take a ribose sugar and make a deoxyribose sugar? And there was one that I talked about earlier today. That's all right, Jordan. Anybody else? Anybody can help? Jordan, Penny? Dorian? Anybody? Do you remember a group? I hope you do. Do you remember this functional group right here? You should be able to say it right there. As soon as I drew that, you should be able to know what functional group I'm talking about. Which one is it? When's our lab practical? It's not for a couple of weeks, right? Which functional group am I drawing? I'm not. No problem, Tulsi. I understand. It's an amine. Okay. So what threonine deaminase does, it takes the amine group off threonine. It deaminates it. Okay. Does that make sense? All right. Okay. So anyway, back to the whole idea of inhibition. So we got threonine deaminase, and again, I'm not going to ask you on an exam to do things like this, but I'm just giving you little hints as you're going through some of these words. I know some of them are unfamiliar to you, and some of them might be a little bit challenging. Like, I have no idea what this means. If you dig into the roots of them, sometimes you can get a little better understanding of what they're talking about. All right, so threonine deaminase is going to be turned into isoleucine, and that takes 5 enzymes. Now, the cell needs 1,000 isoleucine. That's how much it needs. Cell says, hey, I'm going to make a protein. I need a bunch of isoleucine amino acids to make that protein. So make me 1,000. And the cell starts cranking up this machinery that you see in front of you. Bah, 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 bah. And then, of course, the cell says, thank you, that 1,000, that's good. But what do we have up here? Excess, don't we? Right there. We have excess. Maybe another 200 are made. All right, now, it's the excess that make the difference, all right? The excess isoleucine goes back to the very first enzyme, and it binds to an allosteric site. When it binds to that allosteric site, that site away from the active site changes shape. Active site changes shape, threonine can't bind. We call this kind of inhibition feedback inhibition. And so what I mean is it's not any different physically than the non-competitive. This is non-competitive inhibition, of course. It's just the whole idea of, in the overall scheme of the metabolic pathway, the best way to regulate the production of something is to tell what's ever making it when you have too much of it, right? So that's what's happening here. The excess isoleucine, the cell says, I only need 1,000, thank you, that's my 1,000. Those excess 100 or whatever go out, and what they do is they bind to the enzyme threonine deaminase and shut it down and stop it. Now, how do we turn that back on again? Well, sometime in the future, the cell says, hey there, machinery. You remember that isoleucine you made for me? I need another 100. Oh, here's 200, right? I need another 1,000. Here's 200 right here. See, the excess is already there. So the cell then says, I'll take these 200 you got, and you need to make me another 1,000, which, by the way, there's going to be some in excess, obviously. I'm going to take these 200 you got, and I'm going to start using them. And now, since the cell has taken away, see, isoleucine used up by the cell, and now that the cell has used the isoleucine, there's nothing to inhibit the enzyme, and we're back turning threonine into isoleucine. Isn't that cool? It's very simple, isn't it? It's very simple. Mother Nature is all about, maybe you've heard the word before, parsimony. The simplest answer is usually the right one. That's what that means, parsimony. The best way to inhibit the production of something is to use that which you're producing as the inhibitor. Why make another step? Why make the isoleucine signal another molecule, which would signal another molecule, which might then bind to the enzyme? Why do that? Why not just make it the excess isoleucine? You know chemical inertia is going to happen. So use that to your advantage as a living organism. Isn't that cool? That neat? Is that kind of neat little machinery? This is like taking apart the toys when you were a kid. My problem is I could never get them back together, though. Guess that's why I didn't become an engineer. Have you guys heard of parsimony? You know what I mean when I say parsimony? Simple. Simplest answer is usually the right one. You're standing on the beach at night, you look up in the sky, and you see a bright light. You make a hypothesis. It's a star, it's a planet, it's an airplane with its landing lights on, it's a UFO. One of those is least parsimonious. Which one of those is least parsimonious? In other words, which one of those is least likely? Thank you. I was a little scared there for a second. Thank you. Yes, UFOs. I always tell my Bio 2 class, the reason why young children are very bad liars is because they don't understand the concept of parsimony. It is? Not always. Young kids, when you ask them, why did you forget your a jacket at school? It's never, I don't know, I just spaced it and left. I mean, I guess some kids would, but so, I did, but then Johnny needed his, and when Johnny got home, his sister, she took it and put it in the other room, and then, and then on the way out, the dog ate it. It's that. It's like the simplest one is probably the right. That's how you can tell little kids are lying. Right? Because they just make it, this outlandish story. The simplest answer is usually the right one, okay? You'll hear more about that. Always a UFO? I don't think it's always a UFO. All right, I got 3 slides or 5 slides to go, and I can move pretty quick through these not because the information is not important, but you guys are very versed in all of this that's going on. This is a graph obviously. And along the x-axis, the independent variable is going to be the concentration of substrate, that which gets turned into product. So think of this as being the fuel for an enzyme. And on the y-axis is the reaction rate. So on the y-axis is the rate of the enzyme turning the substrate into product. All right, no big deal. You could equate this and say along the bottom is the amount of gas, the pressure on the gas pedal, the speed of the car. It works exactly the same way. So let's take a look at this graph. When I ask you to explain things about graphs to me, please don't tell me the obvious. Like I might say something like, explain the results of the graph you see in front of you. This is what I don't want to see. The rate goes up and then it levels off. You didn't explain anything. You explained nothing to me. I want to know why. All right, so let's start with that. First of all, let's start with the rate going up. As the amount of substrate goes up, 5, 10, 15, 30, whatever the concentration is, why does the rate of reaction go up? Don't make this more complicated than it is. It's as simple, it's as parsimonious as it can be. Oh, cool, I used the word. Why, when I add more substrate to a beaker of enzymes, does the reaction go faster? No one? Why would I step on? Sure. Right? Penny, think about it. It's the same thing as why does your car go faster when you step on the gas pedal? You give it more gas. Substrate is the gas, if you will. The enzyme needs the substrate to act upon. Enzymes sitting in a beaker by themselves are doing nothing. You throw in some substrates, they start turning them into product. You throw in more substrate, they start turning them into product faster. Faster and faster and faster. The more you add, the faster the reaction rate goes. Does that make sense? Everybody good with me on that? More substrate, more reaction rate. Good. Now, can your car, let's just stick with the car for a second, can your car go infinitely fast? If I keep mashing the gas pedal, can I eventually get to what Einstein said, what the speed of light? Can I go infinitely fast in my car? I'm hoping that that's latency and lag, that you guys aren't thinking about this. No, of course not. Penny, you can go infinitely fast in your car. What kind of car you got? Nah, you can't go infinitely fast. Hey, if you decided the other day to run to school instead of take the car, could you run infinitely fast to school? I mean, you could start off with a little bit of a jog, keep going, keep going. Could you keep getting faster and faster and faster and faster on your way to school? Nah, right? Everybody's got an upper limit, don't they? So enzymes turning substrate into product would be like you trying to run infinitely fast. You can't. You're going to get to a point where you can't go any faster, and enzymes do too. So at about 51 of the that's what the little arrow is. About 51 of the substrate concentration will reach something called a saturation point. And after the saturation point, reaction goes no faster. You can add more substrate all you want, but it's not going to go any faster. Enzymes take a little bit of time to turn substrate into product, so they're busy for a millisecond or two. So you can keep adding more and more substrate, and you can end up adding more substrate than the time it takes to turn it into product can happen. So you're going to reach a point where you're not going to get any more product because it's going as fast as it can. If you're ever been a server, you know that's called being in the weeds, right? You can't go any faster. I'm dealing with as many tables as I possibly can. The enzyme's saying the same thing. I'm saturated. You can add more and more substrate all you want, but I ain't going any faster than about 56 or 57 substrates turn to product per minute or something like that. Makes sense, right? Doesn't that make sense? Things can't go infinitely fast. They have a saturation point. All right. Next one should be a little bit easier. So these are what it says up on the top. These are local conditions that would affect an enzyme. You know, we talked about regulating an enzyme. These are conditions that the enzyme would find itself in and how it would react. This is one in which the substrate's changing. Let's take a look at this one. This is temperature. Same thing on the Y. Got it for a second. So we're going from 0, freezing to 60 degrees Celsius, which is pretty hot. 100 is boiling. There, where you see the arrows 37, that's body temp in humans. Once again, don't tell me the reaction goes up and then drops off. I can see that. Let's talk about first why the reaction goes up. Why does the reaction increase as it gets warmer in the environment? Why does that happen? Explain that to me. I'm just going to sit here and wait till y'all do that because I talked about it in class and I know one of you knows it. I'll start calling on you. Christopher, how about you? You got a thought for me? Why does the reaction rate go up as it gets warmer? See, I should make all y'all put your cameras on. Dorian, how about you? You get the right idea. You get the right idea. You got the idea that what made those hydrogen bonds jump and shake around was heat, right? Remember, as you heated up water, the bonds vibrated and eventually heated up enough they broke and it became steam. So you're right when it comes to the fact that heat has an effect by moving things more quickly, right? So again, back to my question, as the temperature goes up, why does the rate of reaction go up? And maybe a sub-question will help. What is it that dictates or causes an enzyme and a substrate to get together? Does it have a peak? I don't know what you mean. A peak, yeah, it does. But it does. But I'm talking about why does it go up? What causes an enzyme on one side of the cell to come into contact with the substrate on the other side? Is it divine intervention? Do they text each other? Is it a train? What do? How do they get together? Well, yes, they bump into one another. That's right. They bump into one another. And one of the ways we can get them to increase the chance of bumping into one another is to speed them up. If they're moving randomly, remember, just moving around randomly. Now, if you make that go really, really fast, you increase the chances of them coming into contact with one another. So back to what Dorian said about heat. One of the ways you can get molecules to move really fast is by heating them up. So the temperature rises from 0 up to about 37 degrees. The rate of reaction goes up because you've increased the chance of the substrate and the enzyme coming into contact with one another. That's all you've done. You've increased the probability. Make sense? What happened there? That's a lot warmer than 37, 60. How come the reaction rate dropped? Okay, here's your thought process should be. All right, enzymes are proteins. Most important thing about a protein, Mr. Clark said, was a shape. Active sites have a particular shape. And then back in chapter 3, we talked about denaturing proteins. And we mentioned a couple things in class that would do that. As it gets too warm, right, Emily? There you go. Just like the egg whites that we talked about, right? As it gets too warm, the molecules are moving around lightning fast, okay? Well, maybe not lightning fast, but pretty fast, but it doesn't matter. You've changed the shape of the active site. You've caused the protein to denature. So bumping into each other, remember, it's a two-part process. You got to bump into one another randomly, and then you got to fit. So bumping into one another is not enough. So at those high temperatures, they're really bumping into one another. Active site has been changed, all right? Now, in this particular drawing, what I've done, the arrow also means something else. We talk about enzymes having an optimum. We talk about lots of things having an optimum, but enzymes have an optimum temperature. This particular enzyme, in my example, the optimum temperature is the temperature with the greatest rate. So that's around 37 degrees before it starts becoming denatured. So optimum, and even on the previous one, we could say that this would be optimum. You shouldn't add any more than 50% substrate concentration because after that, nothing happens. That's what we mean by optimum. All right, any questions about temperature and its effect? So if I ask you on the exam to explain the graph, you're going to tell me two parts. You're going to tell me why the rate of reaction goes up, and then you definitely want to tell me why the rate of reaction goes down. Make sense? So 3 conditions: substrate concentration, temperature, and pH. All right, take a look at this one. Explain this one to me. And again, don't tell me it goes up and it goes down. I can see that. Don't tell me it's got a peak. I can see that. Tell me why it does what it does. This is an enzyme subject to different pHs from 0 to 14. You can see I've already labeled the optimum for you, a little under 4.5. But tell me why it drops on either side of that. So it's like making an egg, and then when all the clear becomes white. No. You've gone too far. Yeah, Dorian, once you've denatured that egg white, you're beyond its optimum temperature. The optimum temperature would have been the temperature the chicken would be exposed to while it's developing and using that egg white as protein. That's why a mother would incubate them, for one thing, keep them at the right temp. But no, getting them an egg is too warm. Anybody got an answer for me? What's happening here? Explain to me why the rate of reaction with this enzyme. It certainly seems that way. Bases do. So do acids, though. But why is that? Why do bases slow down the rate of reaction? Not the rate of substrates, but the rate of reaction, Emily. Why do bases, why do pHs less than 4 and greater than 4 or 4.5? Why do they cause the reaction rate to drop? We talked about it in chapter 3 again, didn't we? What are the things that denature proteins? And enzymes are proteins. Temperature was one. What was the other one? What was the other? I mean, there were chemicals can do it, but what was the other condition that could affect the shape of a protein? Could denature it, for example. Yeah, it does, but I'm talking about in this particular context, Dory. In this context here, is there something on this graph that indicates to you something that might be able to denature a protein? We said heat can denature proteins. Is there anything else that can denature proteins? pH does, right? Acids and bases do. So this particular enzyme, right, has an optimum in the acidic range. Okay, that's fine. But if it becomes too acidic, or if it goes to the basic side, then you denature the enzyme. You change the shape of the protein. You change the shape of the protein, you change the shape of the active site. You change the shape of the active site, no bonding. Okay? Same thing was with heat. The only difference is you don't have that gradual rise. Cooler heat does not denature a protein. That's not why the rate is slower. It's because the molecules are moving more slowly. But here it's more like a mountain peak. You drop off very quickly on either side as you move away from its optimum. No, buffers would be things that would stabilize pH, but it's interesting you mentioned that. Buffers would be necessary to make sure your body doesn't get outside of these optimums, right? If this enzyme, wherever it is in your body, has its optimum at 4.5, and something screws up the pH and gets up around 7, you would want a buffer to bring it back. Remember, buffers stabilize pH. Doesn't mean it stabilizes them as neutral, just stabilize them at the pH they're supposed to be. All right, so thinking about where things are supposed to be, the next couple of graphs are kind of neat because they demonstrate to us this this whole design idea of living organisms and how it's worked through evolution. Look up at the top first, look at temperature. The one in the black is the optimal temperature for a typical human enzyme. Typical human enzyme. Notice what the optimal temperature is too, 37. That's body temp, right? Doesn't that make sense? Doesn't that just like, of course. Why would you make a human enzyme? Why would humans have enzymes that denatured at, say, 20 degrees? Our body temp is normally about 37. So if you had enzymes that were, their optimum was 20, by the time they reach 37, they're denatured. Now, we have no use for the enzyme that are found in the bacteria with the red curve, right? Their optimum is 77 degrees Celsius. 100 is boiling, remember. The heat, many prokaryotes, many bacterial organisms can't stand excessive heat. So we jack up our temperature to and probably for reasons that we're talking about here, it could be that they denature the enzymes. But yeah, the heat in your body is meant to fight off the bacteria. Wouldn't work on this one, though. This bacteria has an optimum of 77. You'd never get your body temp high enough to deal with this one because you'd be dead long before that. But this is a thermophilic bacteria that lives in those hot springs like out in Wyoming, Yellowstone, those geothermal vents at the bottom of the ocean. These guys would have optimums that would make sense to be in the environment they're normally found in, right? So this is what I mean about this whole interplay of the gears of organisms having the conditions they're found in and their chemistry fitting that. Our enzymes would have optimums around our body temp. A bacteria that lives in really hot water would have its optimum much warmer. And down below, an enzyme we use in our stomach would have an optimum pH of about 2. Why? Because our stomach's about 2. Again, it'd make no sense for pepsin. Is that what that is? Yeah. It would make no sense for the stomach enzyme pepsin. Notice not that one doesn't end in ASE. To have an optimum of 4, it'd be denatured in our stomach. And then in your intestines, where it's a little bit more basic, there's something called trypsin. And its optimum would be, a little on the basic side, 8. So I've talked a couple of times throughout class about detective work in biology. So if you were a protein biologist and you were calculating the optimum pH and optimum temperature for an enzyme from some unknown critter, you could get information about what types of temperatures that critter would normally exist in. If you found out that the optimum temperature for an enzyme for some unknown organism was, say, 15 degrees Celsius, just to give you an idea, once again, well, let's make it even cooler than that. Let's say 3 degrees Celsius. So its optimum temperature is 3 degrees Celsius. Would you expect to find that critter in the tropics or in the Arctic? Optimum temperature is about 3 degrees Celsius. Remember, water freezes at 0, so that's very cold water. So where would you find that critter? Tropics or the Arctic? Of course, right? Very good. All right, one last slide, and I'll let you guys go. We hear about this all the time. We need essential minerals and vitamins and all that kind of stuff. And that brings us up to these things that help enzymes along. They're enzyme helpers. We have 2 types. We have things called cofactors, which are non-protein inorganic, versus coenzymes, which are non-protein inorganic. So they're going to vary on whether they have carbon or hydrogen in them. The cofactors are minerals like zinc, manganese. You don't need a lot of that, but you do need some. For example, you need iron. You remember with hemoglobin, that quaternary level of protein structure, it was an iron atom that held those all together. You got to have an iron. Plants, chlorophyll needs magnesium. Did I say magnesium? This is manganese. But magnesium, another mineral, another element, is necessary for holding the chlorophyll molecule together. So you have need for some of these, sometimes in very small amounts. That's why we call them trace elements or trace minerals. Coenzymes are your typical vitamins, vitamin C, vitamin D. Vitamin D, which you get from sunlight. Vitamin D, the active sunlight shining upon your body or radiating upon your body creates vitamin D for you. That's the excuse my wife and I use for laying out at the beach because we love doing it. You only need about 15 minutes of sun to get enough vitamin D for the day. Vitamin B6, vitamin B6 and B12, those are very often associated with those energy drinks, like what is it, the 5-hour energy drink, I think, has a ton of B6 or a ton of B vitamins in it. They give you a little, sort of like a caffeine rush for a little while. But what they do is they help enzymes. So while enzymes make you efficient, these things help enzymes become efficient, these coenzymes and cofactors. All right? Any questions on that? Okay, folks, well, I'm done. That's it for Chapter 6. So that'll, 4, 5, and 6 will be on the exam. Remember, that's going to be next Tuesday. Don't forget your assignments. Your assignments are also doing that Tuesday through Chapters 4 through 6. So make sure you don't miss on those. And if there aren't any questions regarding this, then you guys have a nice weekend. And I will see you next week. And if you have any questions, go ahead and stick around because I'm on office hours for another hour. So we can certainly discuss whatever you need. Sure, Penny. All right, Jordan, you take care. I'm going to stop the recording.